Hello, thank you guys for joining my talk this morning. This is going to be a cell biology talk. So my name is Sabrice Guerrier. I am an associate professor of biology at Millsaps College. Millsaps College is a small liberal arts college in Jackson, Mississippi. We have all undergraduates at our institution. And so all the work I'm going to be presenting today is work done by four-year undergraduate students at Millsaps College. So the title of my talk is Understanding the Regulation of Membrane Curvature During Mating in Tetrahymena Thermophila. And there's a lot of words in there. We're going to unpack this over the course of this talk. So let me give you a brief outline of what we're going to do today. So first, we're going to talk about, we're going to introduce the concept of membrane curvature, really sort of what are the physiological functions and sort of how is membrane curvature achieved. Okay. Next, we'll talk about the role of membrane curvature in cell-cell fusion, or really how we're going to unpack that over the course of this talk. Third, we'll talk about how we're using Tetrahymena as a model for cell-cell fusion and the role of this protein called Reticulon in the process. Next, we'll talk about Tetrahymena Thermophila as a model for studying autophagy, okay? And then we'll finally sort of end our discussion talking about the potential role of this protein called SNX4 in regulating a special form of autophagy called selective autophagy in mating in Tetrahymena Thermophila. Okay, so let's discuss briefly sort of the mechanisms and functions of membrane curvature. So before we get into mechanisms, let's just talk about why we might study membrane curvature. Well, so one of the things that is really important to understand is that cells have to take on dynamic shapes during their life process. And depending on the physiological function of their cells, those shape changes can be pretty diverse. And so if you're a lymphocyte and you need to travel to a site of infection, the cell morphology has to change completely to facilitate the migration of that cell. Well, membrane curvature is going to underlie the shape change that underlies the movement of that cell, okay? We can have changes in membrane shape during the formation of organelles, such as the mitochondria, the endoplasmic reticulum, or the Golgi apparatus, okay? We can also have small changes in membrane shape that regulate the movement, the removal of proteins of the membranes of cells in a process called endocytosis. We can also have the fusion of two membranes, okay? And this is one of the processes we're going to focus on in this talk. So we can have the fusion of two membranes during processes called exocytosis, or the secretion of vesicles from eukaryotic cells, okay? Or we can even have two whole cells fused, and that's the topic I want to sort of unpack in the first part of this lecture. So the process of two cells fusing is called cell-cell fusion, okay? And this turns out to be a pretty important process for a number of physiological processes. One example of an important physiological process is viral infection. So when viruses need to get inside of their host cells, the membrane, the boundary between the outside of that virus and the outside of the cell fuse and allow for the entry of genetic material from the virus into the host cell. You can also have fusion of somatic cells. So you can imagine that two muscle precursors fuse to form a multinucleated myofibril, okay, a multinucleated muscle. And this happens with other forms of tissue, including bone, okay? And of course, the fusion that gave rise to us, right? Fusion of gametes, fusion of sperm and egg, okay? So in all of these processes, the membranes undergo dramatic shape changes. And it's these changes in shape that drive the fusion of these two cells. So how do you shape these membranes. So how are these membranes shaped? Well, there's two ways in which, or two broad categories by which membranes are shaped. In one category, you have proteins that are actually manipulating the membrane. Okay, you can have proteins that embed themselves in the membrane and change the lipid package. You can have proteins that are oligomerized on the membrane. That is, they touch each other and start to shape the membrane as those proteins grow on the membrane. And then you can also have lipids, okay, that can be inserted into the membrane and change the relative package, okay? So it turns out that one of the things that regulates cell-cell fusion are proteins called fusogens that bring membranes in really close proximity, leading to their fusion. But one of the things that my lab has been really curious about is how do lipids play a role in accommodating or shaping that fusion that's induced by proteins? Okay, so what we're really going to focus on, at least in the initial part of this talk, is the potential role of lipids in regulating cell-cell fusion. So lipids can dictate or accommodate fusion based on their geometry. And so if you recall, phospholipids, which make up cellular membranes, have a phosphate head, hydrophobic tails, okay, long fatty acid tails, okay? And it's the relative size of that head group versus the tail that dictates the geometry of the lipid. So you can see in A here, you have a phosphatidylcholine or a cylindrical lipid, which means that the relative size of the phospholipid head compared to the two tails leads to sort of this straight down molecule in the shape of a cylinder. So when these are packed in a membrane, they lead to a very flat sheet. In other words, they lead to zero curvature, no curvature occurring in the membrane. But if you look at the lipids below that, those lipids have a different geometry. They have maybe a smaller head group and one tail, or a small head group and two tails, and those lead to different geometry. So the lipid here, LPC, lysophosphatidylcholine, has a large head group and a single fatty acid table, and is therefore takes on the shape of an inverted cone. And it turns out if you pack a bunch of LPC in a membrane, you start to induce curvature that's determined by the leaflet that lipid is inserted in. So why are we talking about this? Well, it turns out people have looked at the effect of adding LPC into viral fusion preparation. And so what you're looking at here on the y-axis is syncytia formation. So that is a way, syncytia are these big conglomerates of fusion, okay? And on the x-axis, you see the addition of various fusogenic proteins. So HA is a viral protein that will lead to cell-cell fusion, okay? So what you see is that when you add HA, so that's the plus signs, you see an increase in syncytia formation. And this is this can be facilitated by changes in pH. That's not really the point right now. But the point is you get fusion when you add this viral protein, okay? So what happens if you add a lipid that should have some effect on curvature, like LPC? Well, what you see is when you add LPC to the inner leaflet, you block the fusion that is induced by HA, okay? This suggests that the lipids present in a membrane can affect the ability of cells to fuse, okay? So we asked, we were sort of interested in the question of, does the lipid content of a particular membrane, so let me back up and say that what you're looking at here is the exogenous addition of LPC, okay? So our question is the endogenous level, are the endogenous levels of lipids regulated to help facilitate cell-cell fusion, okay? So to get at this question, we wanted to use Tetrahymena Thermophila as a model. So let me set up the model first, and I'll tell you why we think why we particularly chose it for this specific question. So Tetrahymena normally undergo cell-cell fusion when they're in the starved state. So in A, you're looking at two free-swimming tetrahymena. So these are highly ciliated eukaryotic organisms, okay? And these are in well-fed conditions. And what you can see that entire bullet-shaped cell is covered in these hair-like cilia, okay? But in B, what happens is when the cells are starved and mixed with tetrahymena of a different mating type, those cilia are cleared from the very anterior portions of the cell. And those two cells come together to mate, okay? And what's gonna happen in this mating process is there's a fusion pore that's gonna get expanded large enough so that the two mating tetrahymena can exchange nuclei between them, okay? And that's sort of what this crude schema is showing here. So tetrahymena have two nuclei. So you can see in A, you have this really tiny nuclei in black there. That's called a micronucleus, okay? And in blue there, you have a larger nuclei called a macronucleus. So it turns out that the micronucleus is the gametic nucleus. It's purely there to be transmitted during sexual reproduction, okay? That nuclei is transcriptionally inactive, so it doesn't contribute directly to the proteins that will ultimately give rise to the phenotype of that tetrahymena, okay? What it does do is provide the DNA that generates the larger macronucleus, okay? And that macronucleus is transcriptionally active, okay? And therefore will ultimately lead to producing the proteins that give rise to the phenotype demonstrated by the tetrahymena. So what happens during tetrahymena conjugation or the mating process is that micronucleus divides, goes through several divisions, okay? And that's what we're seeing there in B, right? So those two orange nuclei are the results of several divisions that occurred in that black micronuclei, okay? That old micronucleus is destroyed and the products from the division of that micronuclei are exchanged. One of those micronuclei stays in its host cell, okay? And the other copy of that micronuclei gets exchanged with its mating partner, and that's what you're seeing in D, okay? So essentially what's happening at the junction between those two cells is a bunch of membrane fusion and then the expansion of pores that facilitate the exchange of those micronuclei. And so we think we can use that, the fusion events at the conjugation junction, okay, at the junction between those mating types, to get at that question of how are lipids regulating cell-cell fusion. So why do we think we can use that particular event, that fusion event, as a model? Well, there's a beautiful paper published in 2010 from the Ewing group at Penn State, where they used mass spectrometry imaging to look at the lipids that are at the conjugation junction of mating tetrahymena. And what they found was something very interesting. Essentially what they found is that as fusion occurs, okay, as fusion occurs at the conjugation junction, that there was a decrease in the levels of phosphatidylcholine and an increase in the levels of phosphatidylethanolamine. Phosphatidylcholine is a cylindrical curvature resisting lipid, and phosphatidylethanolamine is a cone-shaped curvature accommodating lipid. And so once we saw this data, we really asked ourselves, is this a regulated process? In other words, is the cell actively regulating its lipid content to promote or accommodate fusion? And if so, how is it being regulated? Okay. So one of the ways the lipid content at these conjugation junctions might be regulated is through the activity of lipid transfer proteins that act at the interface between two membranes. And so what you're seeing here is an image of a lipid transfer protein in blue that's swapping out a lipid from the green membrane and adding it to the yellow membrane, okay? So what might be the sites of membrane junctions, okay? So one site of membrane exchange junctions could be probably the most one of the most well-characterized sites are endoplasmic reticulum plasma membrane sites. Okay, part of the reasons these are really well characterized is because people who are scientists that are really interested in calcium-induced calcium release, okay, look at the entrance of calcium from the extracellular space into the endoplasmic reticulum through a bunch of calcium channels that act as these interfaces. Okay, so the point is that lipid transfer proteins could regulate the exchange of lipids at membrane interfaces. Endoplasmic reticulum, plasma membrane are the well-studied and well-characterized membrane interfaces. Okay, and so we were curious if these junctions, these ER plasma membrane junctions, might be involved in lipid exchange in Tetrahymena. And one of the things I should say that's important is that no one's characterized ER plasma membrane contact sites in their role in cell-cell fusion. So in 2015, in collaboration with Eric Cole's group at St. Olaf College in Minnesota, we conducted a study where we wanted to look at the membrane dynamics that occurred during mating in Tetrahymena, during conjugation in Tetrahymena. And so what you're looking at is an electron tomograph reconstruction, okay, of mating Tetrahymena. So in the upper part of that panel, next to the label C, you're looking at one Tetrahymena, and then on the bottom panel of that particular image, we're looking at another Tetrahymena. And that green line there represents the membrane that's being shared by these two fusing Tetrahymena. Okay. So one of the things that was interesting here is you can see that light blue wispy membrane is invading these spaces in the membrane. So these are the fusion pores that result from the fusion of the two membranes. Okay. And the other particular membrane I want to point out here is this membrane right here in blue. Right, that membrane in blue is the nuclear membrane. Okay. And as we started to look through our images, what we noticed was that this blue membrane was also making contacts with the plasma membrane. As you know, the endoplasmic reticulum membrane is continuous, okay, with the nuclear membrane. And so we hypothesized that this wispy membrane that's extending off the nucleus and coming into contact with the plasma membrane here might represent an ER plasma membrane junction. We couple this to some preliminary experiments that we've done using small molecules inhibitors to inhibit lipid transfer, and showing that those small molecules inhibitors in pair formation, we basically came up with a hypothesis. And our hypothesis was that pair formation in cell-cell fusion is mediated by lipid transfer between ER plasma membrane contact sites, and that the formation of these contact sites is dependent on ER endoplasmic reticulum tubulation. Okay, all right. So to begin to test this, we wanted to identify whether there could be proteins that are expressed in Tetrahymena that might lead to the tubulation extension of this blue membrane that we just highlighted. So there are proteins that are very well characterized that are known to tubulate membranes. Those tubulate ER membranes, those proteins are called Reticulon proteins. And essentially those proteins have alpha helices that embed in the membrane of the ER. And when there are enough of those Reticulon proteins in an ER membrane, they can deform the membrane into a tube. Okay. So what we did was we took a bioinformatics approach to try to identify if there were Reticulon proteins that were expressed in Tetrahymena. So to do that, we searched NCBI for Tetrahymena proteins that were homologous to Reticulon proteins in mammals. We then took the proteins that we found in Tetrahymena and ran them through a program called HHPRED, which allows you to compare secondary structure and amino acid identity to known, to actual crystal structures or NMR structures of previously characterized proteins. And so it's a really powerful way for one to do homology studies, okay? So once we confirmed that the proteins that we found in Tetrahymena were indeed Reticulon proteins, we then used the Tetrahymena gene expression database to look to see at what point in the Tetrahymena life cycle is this particular Reticulon gene expressed. Here's what we found. So what you're looking at on the y-axis are normalized expression units, okay? And what you're looking at on the x-axis is the life stage of the Tetrahymena. So essentially what we're looking at here is when during the life stage of Tetrahymena is the Reticulon mRNA expressed. The L here stands for log phase, so these are when Tetrahymena are growing really robustly. S stands for starvation. Tetrahymena need to starve before they can be mated in the laboratory. Okay, so we start. So S stands for starvation, and C stands for conjugation. So this is during the Tetrahymena mating period. And what you can see is that there's a big spike in Reticulon expression very early during conjugation, and this would be consistent with a protein that plays a role in cell-cell fusion. Okay. So we now know that Reticulon is expressed in Tetrahymena. And so now what we wanted to see was, does Reticulon protein localize to the conjugation junction? Okay. So to do this, what we did was we wanted to endogenously tag Reticulon. So we wanted to fuse GFP. We wanted to add GFP to the endogenous chromosome of Tetrahymena in which the Reticulon gene resides. So to do this, we use a plasmid that allows us to clone upstream and downstream flanking sequences of the Reticulon gene next to green fluorescent protein, a promoter called MTT1 that's cadmium inducible, that's driving the expression of a drug resistance marker. And so what this will allow us to do is to select for cells that actually have this DNA inserted in the chromosome by treating it with this drug. This particular gene provides resistance to a drug called carbamazepine. Okay. So how do we get this in the cell? Well, we use a biolistic bombardment. And essentially what this means is we take DNA that we linearize, okay? We coat gold particles with that DNA. And then we can use this gene gun to basically shoot these gold-coated DNA particles into Tetrahymena, okay? And what we hope will happen is that if we flank this particular cassette with DNA near the Reticulon gene, in this case, if we're trying to tag it, the very end of the 3' coding sequence on one side, and then the 3' untranslated region on the other, that we can then add the EGFP, MTT1, and NEO4 to the very C terminus of that particular gene, the 3' end of that particular gene, through homologous recombination. And so our goal is to create endogenously tagged Reticulon using this method. And I'm not going to show you this, but if we just exchange the 3' coding sequence for the 5' UTR, then we can replace the whole coding sequence of Reticulon this way and generate knockouts this way. Okay, so we use this method to generate GFP tagged Reticulon. So here you're looking at mating Tetrahymena, and you can see that there seems to be some in this control cell, which doesn't have GFP, we're getting some bleed through of a nuclear stain into the GFP channel. Okay, that's why you see two dots there that are showing the nuclei. Herx, there's a nuclear stain, so that's actually staining the micro and macronucleus, and then you see the merge there. So these are control conditions, unlabeled conditions. When we look at reticulon GFP expressing cells, what you see is that you see in the GFP channel, you see staining at the right at the junction, okay? And then you also see some nuclear staining, okay, or some staining of something around the nucleus. To see that, let's look at two higher magnification examples. So again, here you see the micronucleus, the nuclear envelope perhaps, or some membrane around the micronucleus being labeled. And then you also see labeling at the junction, as well as these tubular-like structures, okay? So essentially what this looks like to us is that reticulon labels the conjugation junction, so that membrane between, okay, between the two mating cells, and also seems to label some membranes that are staining the very periphery of the nucleus. So this is really consistent with labeling the endoplasmic reticulum, okay? So then what we wanted to do is we wanted to generate some knockouts, some Tetrahymena knockouts to look at the effects of removing reticulon on Tetrahymena mating. So in the top panel here in lanes 1 and 5, you're looking at wild type cells expressing reticulon. In lanes 2, 3, 6, and 7, you're looking at Tetrahymena knockout cells, and you see that those cells do not express reticulon if you're looking at the top panel. If you look at the bottom panel, what we're doing there is we've done RT-PCR for the drug-resistant marker that we inserted in order to generate the knockouts, and you can see that only the knockout cells are expressing the drug resistance marker. So it looks like we indeed have Tetrahymena knockout cells here. So what we wanted to look at with regards to the knockout was if reticulon is at the conjugation junction, and we're arguing that you need ER tubulation to lead to lipid exchange to facilitate the fusion that mediates mating, okay? We wanted to look at what the effect of knocking out reticulon was on the formation of these pairs that are required to form during conjugation, right? So in the top panel here, you're looking at control conditions, and what you can see is you have quite a bit of mating. Okay, so you can see all these tetrahymena existing in these pairs. Okay, but when we look at mating in reticulon knockouts, what we see is a real reduction in the number of pairs. Okay, and we quantify that here. And so what we see here, and I'll just conclude this first part of this talk. So what we see here is that endogenous reticulon labels the conjugation junction as well as the nuclear periphery. So again, that nuclear peripheral staining and some of that tubule staining we saw in those images is really consistent with labeling the endoplasmic reticulum. And it's also really interesting that we also see labeling of the conjugation junction, again, supporting the idea that there may be, these could be sites these could be membrane contact sites. Importantly, when we delete reticulon, we see an impairment in pair formation, again, suggesting that that reticulon may be required for tetrahymena conjugation. So in terms of future directions for this part of the talk, we want to develop a fluorescence marker so that we can actually co-visualize the endoplasmic reticulum and reticulon in tetrahymena. We also would like to perform structure-function analysis to determine which domains on reticulon are required for this function at the conjugation junction. We'd also like to begin to look at proteins that interact with reticulon with the hopes of identifying a lipid transfer protein that could support our hypothesis that lipid transfer is mediating the ability to fuse during tetrahymena mating. Okay. So what we're going to do now is we're going to switch gears to another physiological process that we're really interested in studying in the lab. Okay. And that physiological process is autophagy. So in this particular image, you're looking at organelle shaping as a process that's as a set of processes that are dependent on membrane curvature. And one of the really good examples of something that we think is regulated by membrane curvature is the formation of this organelle called the autophagosome. Okay. So autophagosomes form during a process called autophagy. And autophagy is a process in which the cell begins to eat itself under starvation conditions. So perhaps the best characterized starvation condition is when cells are missing amino acids. And essentially what will happen is that a double-line membrane, okay, will form typically near the ER, but we're seeing now that they can form from a lot of different areas. And essentially what happens is this double-line membrane called the phagophore, okay, will form in the cytoplasm and expand through membrane deformations and membrane fusion around whatever cargo is near it in the cytoplasm. When this membrane expands and encloses that particular cargo, it's called an autophagosome, okay? And essentially what will happen is that autophagosome will fuse with lysosomes, and the lysosomes will lead to the degradation of that material and the repurposing of it for essential functions. So this is the idea behind using autophagy during starved conditions. Well, it turns out that, as you might expect, that autophagy is really is associated with a number of disease states, a bunch listed here. But one of the things that's interesting when considering how autophagy relates to disease is that both the upregulation of autophagy and the downregulation of autophagy suggest that are both associated with disease, but the fact that both the increased and decreased levels of autophagy are associated with disease suggests that there must be some basal level of autophagy that's occurring all the time. And so what we know now is that autophagy isn't only occurring under starvation conditions, but it's part of a maintenance program that's done by the cell. Now, the other function that's arising for autophagy is that not only is it something that's happening all the time, but in some ways it can be weaponized. That is to say that normally autophagosomes engulf things that are near them in sort of a non-discriminate way, okay? But in this weaponization, autophagosomes can be specifically targeted to selective cargo, okay? So in this example here, what you see is that there is a cargo that interacts with a receptor, and that receptor can interact with proteins that are on the autophagosomal membrane. The protein being highlighted here is a protein called ATG8 or LC3. Okay, so there are receptors that bind LC3 and lead to the specific degradation of that cargo. There are great examples of this as it relates to the degradation of damaged mitochondria, expressing receptors that get engulfed by the autophagosome. And so this kind of autophagy, this autophagy of specific targets, is called selective autophagy. And it turns out that selective autophagy has been associated with a number of cardiometabolic diseases. And as I said, Millsaps College is in Jackson, Mississippi, and understanding how metabolism is regulated is something that's really important to this state, as we have a population that's really affected by things such as obesity and diabetes. And so really understanding selective autophagy is important to us in this state. Okay, so as you might imagine, we can use Tetrahymena to study this specific problem as well. And the reason tetrahymena is a really good model for this problem is because the micronuclei, right, those little nuclei that are exchanged during tetrahymena mating, remember, are only gametic nuclei. They are transcriptionally inactive. And so in order for the phenotype of these cells to change under these starvation conditions under which they're mating, they have to make new macronuclei. And the way new macronuclei are developed is by several divisions from the micronuclei that were exchanged between the tetrahymena within this mating program, okay? So, or I should say from the fusion of the exchanged nuclei that were exchanged during this mating program. And so what essentially happens, as you can see in E, is that you get two from the development of the gametic fusion of those, of the two nuclei. What you get is two light blue macronuclei, and those two little pink new micronuclei. And so what ends up having to happen for these cells to gain a new phenotype is that they have to destroy the old macronucleus. And that's highlighted here in the red box. That macronucleus is labeled in green because we know, because I made the cartoon that way. But the reason I labeled it green is because there's been a wealth of evidence in tetrahymena that the old macronucleus that gets destroyed in order for these cells to gain the new phenotype are coated with autophagosomes, okay? And the reason we know that is because there's been some really good work that has showed that ATG8 GFP, ATG8 being a autophagosomal protein, labels these macronuclei, and deletion of a kinase, VPS34, that's required to make the autophagosome disrupts this degradation completely, okay? So the fact that this single nucleus gets chosen for degradation through autophagy provides a great model for us to study selective autophagy of this nucleus, okay? All right, so the approach we've taken to study this is to characterize a protein that may play a role in shaping the autophagosome, and this protein is a kind of protein called a bar protein, specifically a SNCS bar protein, okay? And that's highlighted here in yellow, okay? So why did we choose to focus on the SNCS bar protein? Well, it turns out that in tetrahymena, there are genes that may be co-expressed. That is to say, that there are genes that have very similar patterns of expression. And so when we were characterizing Reticulon's expression, what we noticed was that there was another gene, this gene T-therm 0021930, okay, that had the same expression pattern as Reticulon. And so essentially what we did was we wanted to see if this gene might be playing a role in conjugation, just like we think Reticulon is. So when we looked at this gene, what we found is that this gene has significant homology to a yeast protein called SNCS4, okay? And it turns out that what SNCS4 does is SNCS4 is required for the trafficking of enzymes to the vacuole of mating yeast. And it turns out the vesicle, the transporter that brings these enzymes to the vacuole is essentially an autophagosome. And so the trafficking of this enzyme to the vacuole is sort of a form of selective autophagy. And it turns out that if you knock out SNCS4 in yeast, that you disrupt this really this CVT pathway, or this selective autophagy-like pathway. And so we asked, what might this SNCS4 be doing in tetrahymena? So we generated GFP SNCS4 expressing tetrahymena, and we looked at where they localized during mating. And I'm leaving out some data here for the purposes of brevity. So in this top panel here, you're looking at non-GFP expressing tetrahymena, so wild type expressing tetrahymena. In the first panel, you see GFP. In the second panel, you see lysotracker. And what lysotracker does is track the lysosome, okay? So remember what happens in the formation of an autophagosome. Autophagosomes engulf a cargo, then lysosomes fuse with that autophagosome, converting into autolysosome, degrading that material that's inside the autophagosome. So in the third panel, you're looking at Hurst, and what that's showing is all the nuclei in the cell. And what you notice is that the nuclei at the very back of that cell, okay, are labeled with lysotracker. That nuclei at the very back of the cell is the parental macronucleus, the old macronucleus that must be degraded. So remember now, there are 5 nuclei in each of those cells, yet only one of those cells is lysotracker positive, okay? So again, this is a very selective degradation of this nucleus. When we look at this process when we look at where Tetrahymena SNCS4, the GFP tag version of Tetrahymena SNCS4 is, at this time in tetrahymena mating, here's what we see. We see that in the first panel, you can see that Tetrahymena SNCS4 is labeling that nucleus in the back. That's the same nucleus that's lysotracker positive, and we know it's a nucleus because it's also Hurst positive. And we can see that in this next panel. So what we see is that the lysotracker positive nucleus is the only nucleus that's SNCS4 positive. So what does this suggest? That SNCS4 is associated with the degrading macronucleus, right? The macronucleus that's specifically targeted for autophagy, okay? Interestingly, we see some lysosomes, okay, that are just adjacent to GFP positive, some GFP positive vesicles, suggesting that SNCS4 and the lysotracker arrive independently to the nucleus, which would also be consistent with being autophagosomes. Okay. So we wanted to know if the localization of SNCS4 to this degrading macronucleus was dependent on autophagosome. And so what we did was we looked at whether SNCS4 would localize to the nucleus in the presence of 3-MA, which should inhibit autophagosome formation. So essentially what we're saying is, if there are no autophagosomes, the SNCS4 localize to the degrading, to the macronucleus, the old macronucleus. So in the top panel, you're looking at Tetrahymena SNCS4 GFP. On the first panel, second panel lysotracker, third panel Hurst, and the fourth panel merge. And you can see that Tetrahymena SNCS4 GFP, like we expect, is localizing to the lysotracker positive nucleus in the back of the cell. But when we treat with 3-MA, when we see a reduction in lysotracker in that nucleus in the back of the cell, and we also see a loss of Tetrahymena SNCS4 localization to that nucleus. To see that, let's look at the higher mag image. So again, you can see on the top, perfect co-localization between SNCS4, lysotracker, and the nucleus stain in the back of the cell. And on the 3-MA treated cells, you see a complete loss of localization to that nucleus, suggesting that Tetrahymena SNCS4 is labeling that old macronucleus in an autophagosome dependent manner. Okay. So we generated SNCS4 knockout cells, and we were interested to see if we would see a defect in autophagy of that nucleus, the selective autophagy of that old macronucleus. So in this top panel, you're seeing wild type matings. Okay, we're seeing that nucleus in the back of the cell is lysotracker positive. Okay, we see in the second panel that Hurst is there, so the nucleus, so that is indeed a nucleus that's being labeled. And in the merge, we see co-localization of lysotracker in the nucleus that Hurst. When we do this same experiment in SNCS4 knockouts, interestingly, we did not see a loss of lysotracker positivity in that nucleus, in that old macronucleus. Remember, the idea would be that if SNCS4 wasn't there, then there would be no no autophagosome present on the membrane. If there's no autophagosome present, there shouldn't be any lysotracker, thus no autophagy. And so we were surprised to see that those nuclei were still lysotracker positive. But then we looked at the actual size of the nuclei, and what we saw was that the knockout tetrahymena, the macronuclei in the tetrahymena, the knockout cell, was significantly larger when compared to the wild type cell. Okay, and that's quantified here. And so that was sort of interesting because there's been some evidence that that there are degrading enzymes that are trafficked independent of lysosomes to the degrading macronucleus via autophagosomes that are consumed, autophagosomes that consume mitochondria. And so we're interested in exploring whether this effect on nuclear size is due to a mislocalization of mitochondrial enzymes that end up in the nucleus. So in conclusion, Tetrahymena SNX4 labels the degrading macronucleus. This labeling is disrupted by impairing autophagosome formation, and the deletion of SNX4 correlates with larger nuclei. So future experiments that we want to do. There are additional SNX4 family members. In fact, in yeast, to see robust phenotypes, particularly in mitophagy, in autophagy of the mitochondria, one needs to delete SNX4 and its oligomerization partner, SNX41 and SNX42, in order to see a really strong phenotype. And so we wonder if we're not seeing compensation by the other SNX family members in terms of the lysosome localization. The other thing we like to do is we'd really love to characterize the receptors that are leading to the targeting of the autophagosome. And so one of the things we like to do is use bioID tagging of SNX4 to identify proteins that are in the periphery of the degrading macronucleus. And one future direction that isn't up here is we love to make mutations to SNX4 that would impair SNX4's ability to bind to and/or bend membrane to see if that activity is required for its effects on the degradation of the macronucleus. So I'd love to acknowledge the students that have done this work. There's a lot of undergrads here to name, so I won't name them. But we've done many of these experiments, both in my research lab, but also in some of the laboratory classes that I've taught here at Millsaps. I'd love to thank our collaborators, including the Tetrahymena Stock Center at Cornell University, which helped us to make some of the cell lines that we showed in this presentation. The Turkowitz Lab at the University of Chicago, the Kapler Lab at Texas A&M, and someone that is in here, which is the Ciliate Genomics Consortium, which has been an invaluable resource to me and my students. In terms of funding, I'd like to thank the Howard Hughes Medical Institute for a grant that we had here at Millsaps that is funding a lot of this work, and Mississippi Inbreak Grant for funding this work. I'm going to be taking questions by email, and I really encourage you to email the questions. Well, I'll get the questions by email, and I'll be able to respond to you as soon as possible. I'm looking forward to hearing your thoughts and looking for ways that we can get at some really cool questions